Welcome to the MPS panel at the EMD Electronics booth here at CES. I'm thrilled to be able to bring this to you and show how technologies are converging and looking for disruptive opportunities to transform what healthcare looks like today and going forward into the future. I'll be joined by a few panelists in just a minute, but at first I wanted to give a brief background on Merck KGaA Darmstadt, Germany. So if we could bring up those slides, please. So EMD Electronics is a leading supplier of integrated materials solutions to both the electronics industry, both the semiconductor industry as well as optoelectronics. We have the broadest portfolio of capabilities both for materials and materials-related solutions across the industries. But we are part of something bigger, and this is really going to be the focus of our MPS panel today. We are part of Merck KGaA Darmstadt, Germany, which is the leading science and technology company across the world. And we have three specialized business units: a life science business unit, a healthcare business unit, and an electronics business unit. By combining capabilities, by combining technologies from all three business sectors, we believe that we'll be able to transform what MPS is going forward. We operate under brand names of MilliporeSigma, EMD Serono, and EMD Electronics in the United States and Canada, and then for the rest of the world under the brand name of Merck. So very brief background of the company. Together as one company, we are here to spark discovery and elevate humanity. 358 years of existence. 63,000 employees across the world, 21.2 billion euros in sales, roughly 25 billion US dollars, and then about 10% of that, 2.3 billion euros, being reinvested each year in R&D. By this combined effort, Merck KGaA Darmstadt, Germany, we believe that we have the ability to transform what healthcare is, and we'll cover that in the panel today. So let me invite my fellow panelists up to the stage for our discussion. We'll start with Scott Busby. He's the Executive Director in the Discovery Sciences Organization of Novartis. Welcome, Scott. Peter Peumans, CTO of imec Health. Courtney Sakolish, Lead Scientist for the TEXVAL Consortium. And Haiying Grunenwald, the Head of Biology R&D within the MilliporeSigma Life Science Business Unit of Merck KGaA Darmstadt, Germany. So I wanted to start off with maybe a level-setting question. Organoids, MPS technologies are not necessarily new. They've been around for several decades, at least going back to the 1970s, 1980s timeframe. But they haven't been mass proliferated yet. So what do we see that's holding the adoption back for MPS systems? What's preventing larger scale adoption at this point? Scott, let's start with you.
Thanks, Steve, first of all, for the invitation to be here. It's a real pleasure. I think, for me, at Novartis and all of the other pharmas that we've worked around and we've talked with, I think MPS technologies are really exciting. And clearly, they're driving forward the idea of being able to produce an organ for research purposes that can hopefully help do away with or minimize the need to use animal models to be much more relevant for human disease. I think one of the major issues we've had, though, has been scalability and robustness. In my area, where we do all preclinical screening and high throughput screening for drug discovery, we do thousands of experiments at a time to try to interrogate huge low molecular weight libraries against a number of different reactions and experiments. So we haven't been able to do that reproducibly in these systems as of yet. But I'm excited to see a lot of the advances that the folks up here and around EMD you've shown that are being worked on. So I think if we can work on those two things, that would take us to another level of being able to incorporate this in research.
So scalability, reproducibility. Courtney, the TEXVAL Consortium, you've had a lot of hands-on experience with different systems. What's your take? What's the current state of the art and what's missing?
I was going to say, I could talk about limitations all day. So again, reproducibility is a really big problem, and specifically in biological reproducibility. Using primary cells, using different iPS donors, there's a huge variety of different issues you can run into that, and having a long-term set of experiments that you can benchmark against is a big challenge. Also, in terms of the reproducibility, when we buy a lot of these models, we're buying the plasticware, we're buying the peripherals, we're not buying the model, which includes biology, and that makes a really big difference in how the model is going to perform. Really, the biggest driver in all of the experiments that I've run as part of TEXVAL is the cells you're putting into the model. As long as you have some key characteristics of the chip itself that's going to perform the same. And then lastly, tech transfer. So there's some of these models can be very complex, very difficult to operate. Even the way you load cells, the way you sample, those can really, really have a big effect on how the experiments perform and the data that comes out of it. So having really, really good training, really simple, ready-to-use models, I think is going to be really important moving forward.
And I was just going to add is the adoption is less about the biology, more about the trust, right? This whole organoids, the MPS system hasn't been shown consistently, reliability to outperform the manual models, right? Until then, the pharma companies cannot roadmap and then build drugs based on MPS until that trust is built.
That makes a lot of sense. Peter, what's your view coming from imec? imec is known for 40 years as being world leaders in semiconductor technology, but over the past 12 to 15 years, you've also branched out into the health space in a number of areas. Where do you see this notion of technology convergence going, and particularly as it applies to technologies like MPS?
Well, so we think silicon can add quite a bit in many ways. So in the context of MPS, it's really one of the really important aspects is that you can convince the biology in your system to mimic what happens in a human. And so that requires that you can dictate the architecture of your organ, dictate where the cells go. You want to make sure the cells feel at home in the scaffold that you've created, and we think silicon might be a great way to do this. So that's one aspect. A second aspect in which silicon/chip technology excels is, of course, data. So reading out what is happening with the biology, multiple sensors that give you more information about what's happening with the biology. Today, you tend to be limited by imaging, typically endpoint imaging, very limited other readouts, which really don't give you a lot of information about what's happening inside these systems. So a dense readout, we think, is going to be quite important.
I actually want to add a little bit about the sensors, right? So I think sensors is probably the most important things for MPS to be adopted for actual, pharmaceutical companies and drug discovery. And the sensors we do now is not real time, it's not continuous sensing. So quite often we detect a failure after it's already happened. So speaking of sensors, imec is a world-leading, research programs and institute for the best, most comprehensive, most advanced sensors. I think that's where, bringing all of us together makes that system work.
Thanks for the shout out, Haiying. I also want to add in that context, the data from these sensors is not only important today in terms of understanding the biology, making sure that it's going the right way, but also in generating the data sets that will be required to then educate foundation models that ultimately are going to be quite powerful to add additional insights on the biology, and hopefully in the long term remove the burden of experimentation.
Those are great points, and we'll definitely touch on the hardware and the digital aspects in just a minute. But I wanted to focus on the biology to start. I think the biology, probably all agree, is the heart of the system. Without functioning, repeatable biology, the hardware is not necessarily useful to researchers. So Haiying, your team is within the MilliporeSigma business unit of Merck KGaA Darmstadt, Germany, has done a tremendous amount of work over the years on organoid biology. Can you give us a little bit of a sense of where that stands in terms of technology readiness, and where do you see the remaining development efforts needing to be focusing on that?
So the biological variabilities is at the heart of why NPS hasn't been widely adopted, right? So how do we control the variability? How do we create that consistency? It's three things: inputs, process, and outputs. Inputs, just like you said, cells and the tissues and things that are coming in. And process, how do we QC these cells and tissue? How do we grow them? What kind of a medium? What kind of extracellular matrix that we use to build that scaffolding for organoids, right? Outputs is the data, the analysis, the platform, the machine learning, et cetera, et cetera. So until we address these three variables and make them consistent, reliable, that's across the board, NPS would just be a variability amplifier instead of a decision-making platform and system.
Makes a lot of sense. Scott, what's your perspective? Is a large pharma company really focusing on repeatability of results, being able to bring new drugs to the market requires high confidence in the drug's efficacy? What's your view of where the biology stands today?
Yeah, I mean, that's exactly what we have to have. The confidence that we know reproducibility from well to well to well or chip to chip is accurate. We also want to be able not just to create the organoid biology, but also to perturb it in a disease-specific way, and then be able to rescue those perturbations either through genetic alteration or through small molecule drugs. So that's how we can validate new targets. We can then find those low molecular weight drugs or other modalities to then treat those ailments. I think that the MPS systems are doing a really great job. If you can introduce flow, if you can introduce shear stress or vasculature to actually create cell types and create an environment where they're actually going to do what they normally do in the body, I think that's really important to be able to have that. But you have to be able to know that every chip that you have, that the expression of the genes are correct, the morphology of all the components of the organ are correct. So being able to do that quality is really, really important, or else we really can't utilize it at scale to do drug discovery.
Courtney, what's been your experience in the Texas A&M environment that you do research on for MPS systems, looking at different biology and everything? What's been your experience in the robustness and where things need to go?
Yeah, absolutely. So I think, again, a really big thing that's been touched on is QCing different biology, right? We need a minimum threshold for biological acceptance criteria. We need this to be commonly applied across different models using the same biology. We need a set of known positives and negatives built into each experiment so that you can compare it against different studies. We also need to compare these complex models against simpler models. 2D, where do these actually benefit, a system to be used? So I think, again, having clearly defined criteria for acceptance conditions on these different biomarkers, on responses to compounds, that'll help us with regulation as well and pushing that forward and helping to define a path forward for these models.
So I'll roll the clock forward. We'll say that the QC and the repeatability, the robustness of the biology has been fully validated, fully demonstrated. That's probably the core of a system, but it's insufficient in and of itself for probably mass scale adoption. So let's talk about hardware for a second. Can we give the audience a sense of where the hardware capabilities for MPS exist today, and what would be this ideal set of hardware for how biology and hardware would be co-optimized to interact together? So, Peter, you guys have been doing a lot of work at imec on co-optimizing biology and hardware. What's your sense of this?
Well, so first of all, the systems that are in use today tend to be plastic plate-based systems that have reached a certain throughput, 24, 48, 96 wells. And so some progress has been made in the past, but where the systems are lacking today is in readout. So the ability to read out real-time data is very limited or often absent. Often you have to do with endpoint readout. Endpoint imaging is the only thing you've got. So where we need to take this, we believe, is fix a number of issues. So first of all, we need to make sure that we can put the cells in the right place, in the right context. So making sure there are membranes in place that are essentially invisible to the biology, but allows the cells to organize to mimic a real human organ. I think that's number one. Number two is, as we've already mentioned a few times, sensing is going to be really important. So getting the sensors very close to the cells, to the tissue, we believe is important because some of the markers you're interested in might be short-lived. So you need to sense right where the biology happens. Another thing that came up is flow. It's not just about making the cells are in the right configuration. You also want to make sure they see the same stresses that they would see in a real human organ, and that often that means you've got to mimic flows and fluxes as they appear in a human. So that requires molecular fluidics, and if you're connecting different organs together, being aware of how the fluxes interact between different organs. So those are, I think, key aspects from the hardware point of view. Then from the user's point of view, also hardware related, you want to make sure you come up with systems that are quite flexible and modular, so that depending on what you're trying to do, you can go for simple systems, large scale, and as your questions tend to focus, and you're focusing on a few candidates, you can actually ask more deeper questions, and then you want to start building complexer systems. So the ability to scale in number and scale in complexity using a modular system will be important as well, I believe.
Can I just add a little bit on the hardware, right? Yeah, so I think we talk a lot about sensing, but imaging is incredibly important, right? Until you have an automated system and a box that does sensing, imaging, and also just how to acquire the data acquisition, how to process the data, right? I know we're going to talk about digital next to it, but I feel like right now, all the systems out there, there's a lot of MPS systems out there, and they're kind of like making the biology work for the automation that we have. I think we should think about differently. We need to build automated system, hardware, to enable the biology. Until then, MPS cannot be fully streamlined.
That's a really interesting point. I'd love to get, Courtney, your thoughts. When I was visiting the TEXVAL Consortium this past summer, you had a lot of different systems in your lab, and you've kind of seen what works, what doesn't. And so can you give us a flavor of what do you think the state of the readiness of the hardware is, and maybe building upon some of Peter's points, what works and what doesn't? What would an ideal system look like from a hardware perspective?
Yeah, for sure. So I've worked with over 40 different models as part of the TEXVAL Consortium at this point. So I've really seen every shape, size, level of complexity, everything you can work with. And you guys really took all the good answers. It's having modularity. It's having a known footprint that can interface with existing things in the lab. Also the ability to kind of future-proof. So you want to be able to design off of that and make a more complex model. One of the things that's been really frustrating as part of the consortia is we invest in a system and we invest in a box, and then it works for a year or two, and then the company comes out with the next best thing, and that one's obsolete now. So we have a lot of boxes in the lab, and I think that's another barrier to entry that a lot of companies have, is they just don't want to invest in something that is going to keep changing over the years. So being able to future-proof your model, I think, is going to be very important moving forward.
I actually just want to add a couple of things. So being able to be compatible with existing automation platforms, like for an example, in our group, we have every type of automation and readout technology working with all of our disease areas. So having something where we know, because in this case you're doing very long-term tissue culture, you need to maintain a lot of sterility. A lot of these systems would really benefit and do benefit from automation and adding matrix and adding cells and changing media, adding drugs or perturbations or whatever. So I think being able to understand how that fits in the automation infrastructure that already exists in our current research environments that are non-MPS, I think that would really be very, very helpful to help it be better implemented and integrated into what we're already doing in drug discovery.
Let's pivot a little bit and talk about digital technologies, because as we walk around the CES floor here, we see some amazing, AI-related technologies. And I would say that while digital technologies for life science and healthcare has been there for decades, we're probably only at the cusp of what we can really harness for that. So, Scott, I'd love to get your thoughts on for MPS and then broader. Where do you see the digital needs, the compute and the AI needs for bringing something like MPS to the market where a company like Novartis could actually use it, bring drugs to regulatory approval, and then those drugs to patients?
Yeah, that's a great question. We're all looking at AI and how that's impacting drug discovery. And in all aspects, I know at Novartis, we're looking at this like everyone in pharma and biotech are. How do we use current AI models to allow us to interrogate perhaps fewer molecule starting points to get to our lead molecule, using all kinds of models of protein structure and various things? I think that digital and AI could be very, very useful in these contexts. Certainly when we're trying to figure out what are the protocols that are going to generate the most robustness. For example, in vasculature, I mean, if you can find something that will give you actual blood vessels to an organ, where you can then standardize that, use AI models and different conditions to help you figure out how to get there, and then have that be the standardized process, I think that would really accelerate robustness, accelerate reproducibility, and that would really help. And also in some of the readout technologies that you mentioned with imaging, we know now that bright field imaging using bright light instead of fluorescent probes is AI driven, where you can look at multi-dimensional amounts of phenotypic readouts in a kinetic way without disturbing the system, without influencing it in a negative way. And you can even monitor that over time, get your time-dependent sensing and other things. These are areas that I think AI will really help, and combined with MPS technologies, that will really accelerate, I think, drug discovery. So.
And yeah, data analysis AI, right? I think we can't ignore the challenge of data storage, right? MPS, the imaging in the cells, right? And you have huge amount of data out of each run experiment. How do we store the data? How do we enable the analysis, right? Is analysis happening in local on a computer, which is probably impossible in the future state? You know, how do we make sure that happens in the cloud? When you do have things happen, analysis happen in the cloud, and how do you worry about the data privacy, right? Because there's human data, patient data, precision medicine. So I think we need to address the AI machine learning, address the storage, address the data, privacy, control, regulatory issues if you were to do this in the cloud, which is probably the only way to do it in the future.
It's a really, really interesting point as well, the data privacy and security aspects of things. Peter, I know from my experiences with imec, for decades, imec has been a world leader in novel compute, not novel compute capabilities. What's your view of how the digitization is going to help advance MPS technology and then broader scale adoption?
Well, Scott actually already explained that AI is going to have two main benefits. One is in actually going from data to insights, for example, going from bright field images to richer information. And then two is actually the sensor information that we're going to collect is going to feed these so-called foundation models that ultimately will augment the real physical models. I think that what we've seen is that Haiying, you explained the fear of compute, and there will be too much compute. We're pushing the compute roadmap. We will continue to push the compute roadmap, storage roadmaps. So I'm not entirely sure that it all has to happen in the cloud. I think there will be substantial edge processing capabilities too that we might use. It's all going to depend on how much of the data you want to keep, how much are we going to process. But I see tremendous opportunity to actually augment these physical systems with local compute and cloud compute in these foundation models.
Let's talk for a minute about ecosystems. So problems like MPS and bringing MPS to markets, it's probably generally speaking bigger than one company. At least within Merck KGaA Darmstadt, Germany, we have all three business sectors working together: life science, healthcare, and electronics. So as the African proverb goes, if you want to go fast, go alone. If you want to go far, go together. So how do we see the ecosystem building and playing out? Where are new partnerships needed? Where are partnerships that are working successfully today? What can we learn from those? Haiying, let me maybe have you take a first lead for that question.
Great question, right? So if you look on this panel here, we represent different entity, different corporation, and different focus of our technology and interest, right? I look across the room in the audience. I think you're from different sectors, again, different interests. We all want to compete. We all want to win. How do we build an ecosystem? How do we be a win-win and still all of us have a piece of the pie, so to speak, right? In order to do that, I think for MPS, the entities need to collaborate on the non-differentiated layer and work together and compete on the differentiated layer. What does that mean? Non-differentiated layer standards, what the samples, the QCs, what's the data, what's the metrics we're measuring, and what does good look like? These are the pre-competitive non-differentiated layers. We need to do it together in order to build this ecosystem, to de-risk this ecosystem. We can't do it alone. And what should we compete on is usability, IP, specific applications, and instrumentation, automation, right? So you come together. I think about this coming together, building ecosystem, is building the railroads, right? We need to build the railroads together to have the ecosystem. And how do we compete? Is individual trains. Who gets there first, right? So this is the only way to have the win-win situation.
If I could just add to that, Haiying, great point. So we fully agree. At imec, we are ecosystem builders, and so we're really happy that Merck stuck out its neck to build this partnership that can make a first step towards these ecosystems. Absolutely necessary. And in this case, MPS even more so, because we need to align so many different parties and stakeholders. For example, the regulatory agencies. So we really need to do standards setting. We need to build roadmaps so that as an industry we can move much faster than as if we would be competing with one another without this common roadmap. So we're fully on board.
Can you elaborate on that, Peter? Because I know that imec is basically the world's leader in bringing electronics, particularly semiconductor companies, together, right? It's a who's who of the semiconductor industry that's members of your core partner program. So how have you seen that being built out over the years? What can we learn from what you've seen that's worked and hasn't worked in the past as we would apply this to a technology convergence space like MPS?
Yeah, so we've been in this business for 40 years, as you know. Well, so it's really important, and Haiying mentioned this, that you come up with a model where everybody has the feeling that they can compete. They get something in return from being a part of the ecosystem. You leverage a common standard, common understanding, but yet that leaves enough room for competition. And this is where the imec semiconductor, it's called Semiconductor Industrial Affiliates Program, has worked quite well, because we allow many partners to come together, solve industry ground challenges, and share IP that is non-competitive, but then on top of that, still compete in the market on the products based on that common IP. And so that balance between what is shared and what do we agree to share versus what are we going to keep private, it's important to understand what that balance is. And I think that's going to be one of the key things to focus on when we build out this program as well.
Yeah, and I was just going to add that, obviously ecosystems are important, right? Why do you have to build an ecosystem? So you de-risk adoption. Without adoption, you don't have ecosystem and you don't have any value, right? So build ecosystem to come together, collaborate to de-risk adoption, and then compete to actually capture value, capture the financials.
That makes a lot of sense. So, Courtney, the TEXVAL Consortium is all about companies working together.
I was going to say, you guys are really playing us up. I mean, this is exactly what our consortia does. It provides a safe space for people from different organizations, pharma, government, industry, trade organizations, consumer goods organizations, to come together and agree on platforms to test, on common things to test across platforms, and again, de-risk. You're not having to take this on board, do the training yourselves. You're able to, see the kinds of experiments we're running. We're taking the same biology, same cells, putting them on different platforms and product testing them against each other and sharing that data with the whole consortia. So again, that's that's a common space, and it's been very surprising. There hasn't been a lot of infighting. Everybody kind of wants the same organs, the same standards tested, the same kind of benchmarking. But again, beyond that, you can take that data and then move on to more competitive, testing that you would do with novel therapeutics.
Scott, I'd love to get your thoughts from a Novartis perspective, kind of an end user, customer perspective for MPS systems. What's been your experience moving from more of a buy-sell relationship, more of a transactional relationship, to a partnership or working in an ecosystem? What have you seen that's worked, and maybe in previous examples, or how do you see MPS being developed and scaled, proliferated most efficiently going forward?
Yeah, I mean, I think it's really important, as you all said, to have the same kind of collaboration. At Novartis, we have a very strong internal research component, and we're very proud of our innovation. But we realize it can't be done in a vacuum. And as you said, Haiying, to de-risk, and it's important that we work with people to build the infrastructure, the train track, as you said, because there's a lot of low-hanging fruit to try to, as we've discussed throughout the conversation here, where everybody wins if we can solve some of these problems for reproducibility, scalability, how AI is incorporated in the data and everything. How we then apply, these technologies to unique drug discovery problems. That's where the differentiation comes in, and that's where the different companies can kind of go in their separate ways. But we take a lot of pride in sharing technologies, in developing and beta testing technologies within my group. We've done this in lots of different areas. We have lots of public collaborations that Novartis engages with companies using AI, using other deliveries, because it's all about the patients. It's all about getting medicines as fast as we can to people who need it. And that's really our only goal. I think for me, one of the experience I had was during the COVID times. We had unprecedented cooperation developing some COVID medicines. And it was super rewarding, and talking with other colleagues from competitive companies, we all had a common purpose to try to do something to stop that pandemic, right? And I think that has that attitude's bled over into a lot of these things. We're all excited about technology. And honestly, in MPS systems, I feel, many people feel, this is the next generation to really get transformative medicines to patients. And so anything we can do to share information and help us all drive forward and get those medicines made, I think that's really important. So
I'd like to close. I'd love to get your thoughts about the future. CES is all about looking forward, new technologies that are going to be transformative for people in different forms and fashions. So maybe a quick round robin, a minute or so. We'll just start with you, Scott. Where do you see in the next 5 to 10 years, let's call it 2035, where do you think MPS will be at that point in time in terms of adoption and usage for actually bringing new drugs to patients faster?
Yeah, I mean, I think it's going to be much higher in the flow chart of drug discovery. I think it's going to be incorporated. There's going to be a convergence of not needing to survey, hundreds of thousands or millions of starting points to get to a lead drug, because of the AI technologies that will allow us to do more in silico searching, in silico screening. So then, whether or not the capacity and scalability of these chips ever reaches where we are now with 1536 or whatever, we may not need to get there. And so hopefully we will have the biological relevance much higher, combined with these other technologies, to really allow us to get to lead molecules and medicines and target validation much more quickly. And I think MPS is going to play a big role in that.
Peter, what's your thoughts? What does your crystal ball tell you?
Well, so first of all, I hope that in 10 years' time, there is a truly vibrant ecosystem that allows everybody to move fast, much faster than they could otherwise do. Two is, I hope there is a strong roadmap. It's not just about enabling MPS, but it's about continuing to make it better, more predictive of what happens in the human, so we can get better drugs to the patient. And then number 3, in 10 years' time, this may be somewhat ambitious, but I'm hoping there is some form of measurable success, in the sense that a better drug candidate was was determined, and it translates to an increased probability of success in the clinical phase of development. So I'm hoping for some kind of signs of really making a difference to the patient ultimately.
I love that. Love that. Courtney, Texas A&M university environment, you're probably at the tip of the spear in terms of where the research is going and where the state of the art is. What do you see the next 5 to 10 years looking like?
Yeah, so I mean, I'm hoping we'll have some really good examples, like Peter mentioned, where the decisions, real decisions, are being made using these models. But again, it really depends on having biological criteria. It really depends on having some kind of incentive from regulatory agencies to be using these technologies in submission packages. We really need push kind of from all ends to kind of move these forward. So I really hope that in the next 5 or 10 years, we're going to have not a human on a chip. You know, I don't think that's going to happen anytime soon, but maybe a couple really good examples for specific contexts of use where these models can make a real difference and can differentiate from 2D and from animal models. So fingers crossed.
And Haiying, I'll let you bring us home here. MilliporeSigma, is going to be a provider of MPS systems. Where do you see the next 5 to 10 years going? What would you like 2035 to look like?
So in 5 years, MPS should be performing better than the manual model, right? It should replace that. So it should be becoming a standard operation for pharmas and for drug companies to use to develop drugs and new therapies. In 10 years, it should be incorporated in just a scaled standard R&D work, right? So when MPS is made is when it's not whether to use MPS, it's when and how to use MPS to make decisions, Ultimately, this is how things get done. If we continue progress, precision medicine relies on things like MPS in order to move forward and advance.
So what I'm going to take away from our conversation here today is that the biology needs to be robust and repeatable. The hardware needs to have data-driven insights, sensors, and modularity. A digital component's got to be an additional layer to enable ultimately in silico experiments to be conducted and new insights to be coming from the data. And then ultimately it's going to be about more than just one company. It's going to be building an ecosystem and how do we make this all happen together. So thank you so much for your time and insights here today, and I wish you all enjoying the rest of the show. Thank you.
Thank you very much.